Hemophilia B is an X-linked recessive genetic disorder that arises from a mutation in the factor IX gene. This mutation prevents the production of the blood clotting protein, factor IX. As a result, patients with hemophilia suffer from spontaneous and injury-associated bleeds that can be debilitating and in some cases fatal. Gene therapy treatments aim to replace the faulty gene with a correctly functioning gene using a virus called AAV8. AAV8 is an adeno-associated virus, specifically chosen for hemophilia gene therapy due to its low prevalence in humans, resulting in a smaller percentage of patients having prior immunity. The AAV8 vector was engineered to contain three plasmids, one containing the functioning factor IX gene, another containing modified viral genes, known as rep and cap genes, which create the outer protein shell of the virus, and a third containing adenovirus helper factors that allow the adeno-associated virus to infect the liver cells of the patients. These three plasmids were then placed into a eukaryotic cell, which acts as a virus-producing factory, creating the outer protein shells of the virus, which are known as capsids. The plasmids that are within the cell then migrate into the capsid, creating the AAV8 virus, which is then released from the eukaryotic cell. These genetically engineered AAV8 viruses were then injected intravenously into the hemophilia B patients. The virus has a strong tropism for the liver, meaning that the AAV8 virus will naturally migrate to the liver and begin to infect the liver cells. Upon reaching the liver, the virus infects the cells using the rep genes and the adenovirus helper factors from the plasmids. The factor IX gene is then inserted into a specific site on chromosome 19, named AAVS1, allowing the cell to start creating the factor IX protein. However, the gene must be double-stranded before it is inserted into chromosome 19. Adeno-associated viruses only carry single-stranded DNA, and so a second strand has to be synthesized for the factor IX gene before it can be read by the liver cell. To combat this problem, researchers have created a factor IX expression cassette that allows the gene to become double-stranded without the use of additional helper viruses. The factor IX expression cassette was engineered to contain the factor IX gene and a complementary factor IX gene copy, separated by an inverted terminal repeat, which is mutated by the deletion of the terminal resolution site. When the gene is released into the cell by the virus, the mutated middle inverted terminal repeat begins to fold due to base pairing, which continues throughout the gene. The complementary factor IX gene folds over and base pairs with the factor IX gene, creating a double-stranded DNA sequence. The factor IX gene is then integrated into chromosome 19, allowing the cell to begin production of the factor IX blood clotting protein. Through this biotechnology, hemophilia B patients can start to produce their own treatment, improving their quality of life.
Created using Powtoon.